Okay, hello everyone, and welcome to today's webinar from BioInformatics, Cutting Edge Trends in Synthetic Biology. I am Keely Trull, Senior Marketing Manager for Science and Medicine Group, and before we get started, I wanted to cover a few housekeeping items. First, if you experience any issues throughout the broadcast, please feel free to chat them in and I'll work to help you fix them. Second, this presentation will be available on demand after the webinar has concluded, and you'll get a follow-up email with that information, and it'll tell you how to access the recording. Third, we will be taking time to answer questions at the end of the presentation, so please submit your questions to the Q&A section, and you can do that at any time throughout the presentation. Leading us in today's discussion is Emma Britton. Emma is a Science Advisor and Qualitative Lead at BioInformatics. Emma, thank you so much for joining us today.
Hey, thanks, Keely. Hi, so I'm Emma. So today I'm going to be talking about this project that we did on, again, synthetic biology, and we were actually able to talk to a lot of folks in this area and learn about where they saw this field going. So we're going to start off and talk a little bit about how we accomplished this study, and then go into some of the key areas that we talked about, and then talk to you a little bit about what additional insights that would be offered in the full report. So just to talk about how we actually accomplished this study, because it's a little bit different than anything you might have seen from us probably before. So bioinformatics, we're part of the Science and Medicine Group. We did in-depth interviews with key opinion leaders from the synthetic biology space. So what we did was we created a discussion guide that was meant to make it so that we could talk to these people and really understand a couple different areas. So our objectives were to define synthetic biology from the people who are actually conducting this kind of research in the laboratory. We wanted to identify the companies and brands that are supporting people working in this research, with a special consideration towards understanding the companies that are creating synthetic DNA and providing this to researchers. And we also wanted to talk to these people and understand where they saw this field progressing, both in terms of the kinds of research that are happening and in terms of how companies and brands can support this research better. So once we had a discussion guide put together, these interviews were carried out by moderators at BioInformatics that had STEM backgrounds, so that we could really establish a rapport with the people that we talked to and have deep, really meaningful conversations with these people and probe into the areas that we found really interesting and get really close to understanding what these people were doing, the areas that most interested them. So these were not formulaic conversations, and some of them really did go off in some really interesting tangents. And we fielded this to the Science Advisory Board, which again is part of the Science and Medicine Group, as well as finding some additional qualified life scientists. These people qualified for this by having a self-identification of working in synthetic biology for 4 years. Because this is a new field, we really thought that that was enough to make it so that these people were well qualified. And they worked in a variety of areas, either biotech, pharma, or academia. And what we did was this led to literally hundreds of pages of transcripts, as well as call notes. And so we took all of that data and we put them together and created the report, which is available from the Science and Medicine Group, as well as this webinar. So I want to talk a little bit about qualitative research because it's different than quantitative research in some important ways. So since we talked to an N of 18 people, this is a small sample. It's pretty big for a qualitative study, especially in something as niche as synthetic biology, but it's important to know that these are specially recruited respondents and that they didn't all get spoken to exactly the same. You know, we tried to go down specific interesting areas with these folks. Even though we had that discussion guide, it wasn't meant to be a script. So it's important that even if we have charts and graphs that say 40% of respondents said X, Y, or Z, it should be considered directional rather than conclusive. Also, we provide verbatim comments in this and in the full report, but we do tend to edit those a little bit to make them a little bit shorter, or for particular grammar. Again, these people were speaking off the cuff to us, and so sometimes, obviously, they say like or stutter, have pauses, things like that. So we do edit those a little bit, though we try to be very sensitive to make sure that it is the meaning that the respondents had. And we also are very careful with the attributions from these, because we want to make sure that the people who talk to us feel that they have their confidentiality protected, and that we never ask them anything that could be considered confidential or proprietary, but that they feel comfortable coming back and talking to bioinformatics again and again. So who did we actually talk to for this? We had a total of 18 respondents, 67% in biotech or pharma and 33% in academia, and they represented folks in the US as well as English-speaking areas of Europe. In biotech and pharma, we had a total of 12 respondents. They ranged from staff scientists, PIs, and a couple of directors as well. So we had people who were directly on the bench and then those who had a broader view. I think looking at these companies, it's also important to see that we have some really small startup areas, biotechs that are in sort of the more Series A, Series B, very small space, and then some of these more established brands. So I think we were able to have a very broad view of what folks are doing. In the academic profile, we spoke to PIs, research scientists, as well as one postdoc. So I think that gave us a really nice view of how some sort of younger academics, as well as more established academics, were able to see this field. So first, I'm going to go into a brief summary of what we saw in this report, and then we're going to go a little bit deeper. And I want to remind everyone that throughout this, if you have any questions, you can drop those in the chat on the webinar, and I'll address those at the end. So I think one of the biggest takeaways from this area is that synthetic biology is a rapidly growing field. It's extremely popular, and it has a great diversity of people partaking in it. So they're coming from all different fields of science. It's very rare for people to actually have degrees or formal training that says synthetic biology, but they're all, sort of brought together by this idea that they can create novel biological function that will solve industrially relevant problems. And when I say industrially relevant, I do include healthcare in that idea. So one of the first big takeaways we had is that synthetic biology is a new field that's similar to genetic engineering, but it differs in many interesting ways. And this is a space that I had sort of assumed was a difference that I just hadn't understood because this wasn't my immediate academic training, but it really did end up being something that even the most sought after academics who taught classes in this sort of rolled their eyes about when I said, tell me the difference between synthetic biology and engineering. It was really interesting to hear that, all of these folks sort of differed a little bit on this. And so it was really interesting to talk to people and see the key differences and similarities between these two fields. And we'll talk about that a little bit more later. But we saw that the scope tends to be different, rather it being a systems-based approach or a targeted approach, and then sort of how you're going about it, whether it's whole pathways within an organism or individual nucleotides or genes. But the assays that you're actually running, or the kinds of tools that you're using, your sort of biology toolkit, are often very similar for both synthetic biology and genetic engineering. When we asked about how people were actually carrying out this research in the laboratory, we saw that outsourcing is becoming really very popular. And it was interesting because there were sort of two big funnels for why outsourcing is popular. One is because it can provide specialized knowledge or equipment, so in terms of experts, but also in terms of very expensive or specialized equipment without having to spend that capital expenditure. So that's very important for small biotechs who maybe don't have the resources for that, or large biotechs who don't want to be hiring people for these specific roles that maybe they don't need long term. We also saw that contract research organizations, or CROs, are acting as experts on demand for more niche services. So things like FDA regulations, having GMP services. But also this other funnel is for more routine areas. So things like sequencing, which used to be a very niche service, but is now something that we sort of see as an everyday function, but maybe it's something that can be done cheaper at scale. So we'll talk a little bit about that in a moment. And in terms of trends in this area, everything that we heard in terms of where this area is going, the kinds of skills that people will need, it's all about having this overlap between computation and biological. So being able to understand what is going on computationally, being able to do the sort of more in silico approaches, and then being able to understand the genetics. These computational skills are becoming more and more important for all of the future applications, especially because multi-omics approaches are becoming more and more important. No one is just running NGS anymore. No one is just running an RNA-seq. We're seeing all of these approaches becoming important throughout the whole process, and being able to look at that data and have a way to de-silo it is increasingly important. And having manufacturers who can work with you in that process is extremely important. So let's get a little deeper into these areas. So when we asked what is synthetic biology, I thought it would be a simple question, and it really wasn't. So our experts differed on an exact definition, but this, I think, really highlighted that this field is coming from a lot of diverse academic disciplines and is also rapidly evolving. So all of our respondents agreed that this is a kind of cellular or metabolic engineering, but whether some of them define this as just a rebranding of genetic engineering, because genetic engineering really does have a bad rap in certain circles, or whether this is a new approach was something that was often debated even by individual respondents. But we did see that synthetic biology tends to be more global rather than targeted, and that it focuses on entire pathways rather than individual components, and that the scope tends to be different. But as I said before, we're using very similar tools like CRISPR, NGS, bioinformatics, and other omics tools, and that for both genetic engineering and synthetic biology, we're going to really need recombinant DNA technology. And the scale of that is increasing pretty exponentially, which we'll talk about in just a second. So how is this being actually carried out in the lab? What kind of tools are important? And one of the areas that we highlight in this report is this need for outsourcing. So when we outsource, we're using outsourcing for specialized equipment or expertise. And CROs are becoming increasingly important, and we saw this huge surge in CRO usage during the pandemic, which doesn't seem to be slowing down. It's definitely cheaper to outsource in many places, and it can add expertise that your company doesn't have. So if you have an area, an assay that you think you're not going to be running regularly, it can allow you to augment what your company is already using, and also allows you to bring in things that perhaps you don't want to keep on hand. So they're doing it routinely. They have the reagents. You don't have to worry about bringing in a whole lot of things and testing them, validating them, and then also keeping that on hand and maybe having those expire. So if it's an area that's not your focus and you're just using a particular assay or test to do some validation, it's an excellent area. It's also something that folks are using because of specialized areas of knowledge such as GMP or FDA regulations, but this is an area that definitely needs more growth. So we had several people complain about not many companies existing that understand or have the capacity to really work with specific FDA regulations or be working under a GMP or GLP regulatory environment. And so this is an area that definitely needs growth. Outsourcing is also something that is preferred for things that can be easily accomplished by an outside laboratory that maybe doesn't have to be as bespoke. So sequencing services are something that were once very much considered something that a core on your campus or that would be sent out as a niche. And then, more recently, it was something that people said, oh, the price has really come down. Let's bring these back into the lab. However, these are often now being outsourced again because it is so cheap. And so we had a lot of people say like, oh yeah, we brought that in-house because the price had come down, but we're actually going out of house again because it's become more cost effective, especially with these huge machines like the NovaSeq. It also allows people who maybe don't have a consistent workflow or a consistent output or throughput to be able to use it if they don't have enough material to justify starting a whole new chip. They can run just a few samples at a time, and that's really helpful so that they don't have to be able to completely fill a chip or, again, to be able to keep those expensive reagents or chips on hand that might go bad. Another area that's very popular in outsourcing and that we go pretty deeply into in this report is gene or oligosynthesis. So no one that we talked to had ever worked at a company that made their own oligos or DNA. This is something that is always outsourced. There are very popular companies in this space that are doing it. The thing that is new and interesting is that usually people would procure their oligonucleotides and then do things with those, put those into their viral vectors, make their plasmids. These are things that can now be outsourced and are becoming popular. So whether it's ordering them at the plasmid stage or actually ordering whole long synthetic gene constructs and not doing any of that in-house is pretty interesting and is becoming more and more popular because it saves a great deal of time and also means that you don't have to have some of that equipment or staff that would do that previously. So where did these folks see the future of this area going? A lot of people saw that synthetic food or protein sources is becoming really popular. We heard a lot about synthetic meat. I think that this is something that people are really excited about and seeing that as maybe one of the first big wins for the synthetic biology space. We also heard a lot about synthetic milk. Environmental applications are something that I think is really exciting for people in this space because I think it really offers some hope to people as well as just an area that has a great deal of industrially relevant areas. So creating organisms that could capture CO2 or stop the bleaching of corals. So areas that you can tweak an organism so that they can be better in terms of breaking down plastics or breaking down petroleum products. So that was an area that a lot of people talked about as a very soon future application. And then of course, novel therapeutics. So being able to engineer small molecules, biologics in this space for new pharma products or even new kinds of molecules that haven't been used before. But when we ask people about how this will all be carried out, who will be the synthetic biologist of the future, everyone really talked about the ability to work with large amounts of multiple kinds of data and make predictions about these genetic constructs. So it was extremely popular to talk about having this deep understanding of both basic biology and computational work, and that this is becoming this very popular hybrid breed of scientists, that you have to have a foot in both worlds, which is becoming a little rare. We seem to have people specialized in one side or the other, and that being able to bridge it starts to become very popular. So I think this was really important to notice, that having the ability to look at the computational data and to be able to understand the genetics is something that is irreplaceable in this field and will become more and more popular in the future. So what else is in the full report for this study that I didn't cover here? We talk a lot about brand considerations for custom genes and oligos. We go into individual brands and what people liked and didn't like about them. We also talk about pain points regarding these custom genes and oligos. We also asked these folks how they learn about synthetic biology, what they like most in terms of, journals, conferences, things like that. We go in a lot more to understanding the skills for this next generation of biologists and how people will need to understand things to be successful in the future in this field. And then we talked a lot with people about how brands working in this space can increase loyalty, how that they can serve this next generation of the hybrid computational and genetic synthetic biologist and be better partners to this area. And I think this was maybe a very fascinating topic because I think that brands need to understand that most of this work is not a set workflow, that it's all going to be bespoke. And so the more that they can work with these folks and really understand the science that they're doing, the more they can increase that loyalty. And we have several slides talking about that. And then the rest of the report just has a ton more verbatims from this space because we have 18 hours of interviews here. So bioinformatics does these kind of studies. We do them custom for brands, the qualitative as well as quantitative, where we survey people from the Science Advisory Board. I'm, again, the head of the qualitative team lead. I love talking to scientists all the time. It's my favorite thing. And we also have more similar reports to this. So if you click here, everyone after the webinar will get a copy of this. And you can see more of our off-the-shelf reports as well as get in touch with us if you're interested in doing custom research, either qualitatively or quantitatively. And there's a couple more recent bioinformatics reports from our quantitative team. We have a really fascinating one about how life science professionals perceive artificial intelligence. And we also have one about perceptions of environmental sustainability in the life science. So we've got about 6 minutes left. So I really appreciate everyone coming here and listening to me talk about this area. I found it really fascinating to interview these folks. So does anyone have any questions? You can drop those in the chat.
Thank you. And just to follow up, Emma, that was a great presentation. Thank you so much. If for whatever reason we don't get to your question, we will follow up with you after the webinar is ended over email. So we do have one in the chat already. It says, thank you for taking this webinar. What kind of things are included in outsourcing sequencing services? Does this include analyze data and provide report?
Yeah, so this is actually something that we talked about a little bit with these folks when we talked about sequencing services, is that one person mentioned that so this was an academic, and they mentioned that their core facility, like their academic core, which is how, I have done sequencing before in an academic core, no longer existed because it just wasn't being used as much anymore because it was so much cheaper to go to CROs now. And they mentioned that they had sort of previously seen, and remember, this is qualitative, so take everything with a grain of salt. They had previously sort of imagined that all CRO-based sequencing services were mostly identical, and that they had learned that they really weren't, that different levels of service really exist within the CRO community, so that you can have something where you just send your DNA or your RNA that's already been extracted, and they will send you back whatever level of coverage you're asking for, and you'll just get your FASTA files. Or there are more bespoke services that can, you can send your raw tissues or whatever, and they'll do extraction and sample preparation and library prep and all of that. And that there are bioinformatics services that are included as an add-on. And so I think that there are different, and I'm obviously not trying to advertise CROs, right? But I think there are a lot of different ways that people can work with CROs. And I think that's very interesting and maybe not as well known to smaller biotechs who are sort of new to working in this space, especially with how popular CROs have become recently. One of the other things we heard from a really large organization was that they contract a bioinformatics company to sort of run all of their bioinformatics on a sort of consulting basis. So they do their sequencing in-house, but they have a bioinformatics company doing that. And so I think it really runs the gambit of all of that. And it is something that is definitely seen as a need. We had another person mention that, like, hey, you know, we're a smaller organization, or we had another person who was like, we're an academic, and we would really like if that was sort of more available on a freelancing consulting basis, because, you know, a lot of people can find a bioinformatician who understands NGS data, but we're working with spatial transcriptomics now, and we don't really know how to deal with that data. So it would be great if freelancing was available in this space. So hopefully that answered your question.
Great, thank you, Emma. Any other questions from anyone else? We have a few more minutes if you want to drop those into the Q&A. Okay. All right. If there aren't any other questions, that'll go ahead and conclude our webinar for today. As mentioned, if you have any follow-up questions that you think of after the presentation is over, you can email us at info@bioinfoinc.com. Thank you all so much for joining us, and I hope everyone has a great rest of your day.
Thank you.
Thanks, everyone.